So today we want to look at how we diagnose malaria from the laboratory. Malaria infection is malaria infection is caused by a hemoparasite called Plasmodium of various species. But here in Africa, we basically have different techniques we use for diagnosis. One of them is using a rapid diagnostic kit, which is called a malaria RDT, a rapid diagnostic test. This is a test kit which has a cassette inside, and you're able to run your blood sample through it. It is embedded already with an antibody. It is embedded with an antibody that when you add your blood, which contains an antigen, it's able to capture it. So one of the methods we use to diagnose malaria is through using a rapid diagnostic test kit for malaria specifically. Secondly, we usually use a slide or a smear, malaria smear. You can either have a thick smear or a thin smear if you want to diagnose malaria. So I want to take you through briefly, practically here, how you can diagnose malaria. So what do you need? You need your pair of gloves. You need your cassette malaria RDT. This one is specific for all the species. Okay, it does all the 4 species: falciparum, vivax, oval, malaria mainly. So we want to see how we can do the test. You have your gloves on. As a safety precaution. And then you should have a sample dispenser. You mix your sample properly. You pick about 5 microliters of blood from the sample dispenser, and you have 2 windows here, 2 dispensing points. The last one is always for our buffer. So after dispensing your blood, you're able to add a buffer at the buffer point here. Depending on the SOP from the manufacturer of the kit, I've put 3 drops. So you leave this test kit to run for about 10 minutes, 10 to 15 minutes. And it will give you your results. For the smear, you can make a thick smear. A thick smear takes quite a large amount of blood compared to a thin smear, but mainly because you want to concentrate parasites, the Plasmodium parasites. So you add your smear in the middle of the slide, like that, and you spread it in a circular form. To this diameter. Okay? So when you have your smear, not too thick, not too thin, it can be translucent. This is a standard thick smear. Not so wide, but in the middle of your slide. Okay? If you need a thin smear, because if the test comes out positive, you might have to do quantification and also identifying the species. Then you also need to use a thin smear. Just like last time I showed in the previous video, you need a thin smear to make your thin smear for quantification. So you can use the same glass slide or a cover slip, and you spray your smear. Okay? So you have your thin smear and the thick smear. These slides should be labeled by the patient's number or patient ID and the date. That patient ID and the date the same. You have the patient ID on your RDT and the date. So, even before the test is done, you are able to see that the rapid diagnostic test kit is already showing a reaction. At the region of PF, this is Plasmodium falciparum and the other species. This, at the point C, it's where the control line is supposed to appear. So when you have two lines, it indicates that it's a positive RDT. If it's a three line, it means it covers all the other species, the falciparum and the other three species, vivax, ovale, and malaria. So these smears, you leave them to dry, and after drying, you'll be able to stain them. You'll use this to detect whether true they are Plasmodium parasites, and you'll use this thin smear to quantify them, either in percentage form or per the number of WBCs you have seen, or you can quantify them per high power field, depending on what you're comfortable with. So we'll wait for our smears to dry, then you fix the thin smear and stain it with either Leishmania Leishman stain, or you can use field stains, or you can use Giemsa stain. These stains always bring out the best picture of the parasite.